Hello everyone. Unit 12 is considered to be one of the least important unit from CSIR point of view. But still it holds relevance as questions are being asked from this unit as well. So let's delve into a few examples of such question for a better understanding. But before going ahead, just a reminder that from this unit you can focus on gene transformation by agrobacterium, from where you can always expect a 4 marks question. Apart from this, other topics, it will be like a rotational basis. Sometimes they ask, sometimes they not ask. So in today's session, I have picked up the questions from certain subunits from Unit 12, so that you will have an understanding what kind of questions are being asked from those units. So let's have a look onto that. The first question is all about gene transformation by agrobacterium. So they have provided with 4 statements: production of single-stranded tDNA by Vir D1 and D2 proteins, interaction of Vir E2 with VIP1 and Vir E3, use of Vir B Vir D4 type 4 secretion complex, activation of Vir A Vir G complex. Now you need to identify the correct sequence of events that will occur during the gene transformation from the bacteria into the plant cells. So as you can see, the correct sequence is DACB. That means virulent A and G will be activated first, tDNA will be synthesized, they will be interacting with some group of proteins, and finally via a conjugational port they will be transported to the plant cell. So attending these kind of questions requires 2 things. First is that you should be clear about the functions of each proteins. And second, the sequence in which the proteins are activated. So if you look into this table, we have a number of virulent genes involved in the gene transformation experiments. Not in the experiment, in natural process as well. Virulent A gene sends acetoserengon that is being secreted by wounded plant cells. So that is the first virulent gene to be activated within the bacteria. Vir G is a transcriptional activator of Vir box. Vir box is something that leads to the activation of other virulent genes. Vir D2 protects the 5' end of the tDNA from being cleaved by exonucleus. So basically its function is protecting the 5' end. Vir E helps in the gene transfer. C1 and C2, they will form overdrive sequence, and that will also help in DNA transfer from bacterium to the plant. Vir D is responsible for excising the tDNA. Vir B, it forms a conjugational port through which the transportation takes place. Of course, for the movement of DNA, you require some input of energy that is provided by Vir B11, ATP's activity. And finally, we have Vir H that will detoxify any molecular components that may come into the transportation process, that is during the DNA transfer. Okay. So, how exactly this gene transformation takes place? This is our plant cell and this is the bacterium. So, when the plant is injured, it will release some chemicals, and that chemicals are known as plant signals. It will be in the form of sugar molecules, certain phenolic compounds or acid. That will be received by certain virulent genes like virulent A, G, chromosomal virulent genes like E, G and I, which in turn helps in the phosphorylation of virulent G gene, thereby activating it. This virulent G gene is responsible for activating other virulent genes. Now, this is the type plasmid, that is tumor-inducing plasmid, from where the tDNA has to be extracted. And towards the 5' end where D2 will be bound so that it will be protected. There will be a conjugational 4 or the type 4 secretion system made up of Vir B and Vir D4. Accompanying this tDNA, we have Vir E2 and Vir F that will be released into the plant cytoplasm. From there, the corresponding tDNA will enter into the nucleus of the plant cell via nuclear pore complex. So before entering, there are some host factors present that will help in the release of this extra proteins or the genes, that is virulent genes. Extra in the sense the helping genes. And finally, the tDNA will be released into the nucleus, gets integrated within the nuclear genome. This is known as a kind of recombination. We call it as an illegitimate recombination, which is known as non-homologous recombination. So if you are clear with the virulent genes and their function, you will be definitely able to click on answer from this question. Next, let's look into the second question that comes from plant tissue culture. Somatic embryogenesis is an important exercise in micropropagation and genetic engineering of plants. Following steps are considered as critical for achieving somatic embryogenesis. Reducing the concentration of sucrose in the medium by half, addition of hormone like 2,4-D to induce somatic embryos, reducing the agar concentration to 0.6%, use of maltose in place of sucrose as a carbon source. So you need to identify the correct combination of statements here. So the correct combination is statement B and D. Sucrose is an important constituent when it comes to plant tissue culture. So if you are reducing its concentration, definitely the growth will be affected. 2,4-D is an example of a synthetic hormone, synthetic auxin, I can say. And this, in conjugation with other plant growth regulators, helps in somatic embryogenesis, specifically in the initial stage. Agar is required for support of growth. So if you are reducing the agar concentration, that support will be lost. Then coming to maltose, yes, maltose can be used in place of sucrose as a carbon source. Let's have a look into that, how and why. First thing is that we all are aware sucrose plays a major role in somatic embryogenesis because it serves as an energy source, it is an osmotic regulator, it is a carbon supplier as well as inducer of organogenic responses. And its presence is crucial for the support for growth, development and differentiation of plant tissues during the in vitro propagation or genetic manipulation processes. So while both maltose and sucrose can support somatic embryogenesis, but when we take the case of maltose, it has certain advantages. For example, the efficiency is better, reduced osmotic stress, and there are chances that when we are using sucrose in the media, browning takes place. So maltose reduces the risk of browning, enhanced differentiation, stability in culture as well as genotype specific responses. So the specific effect of maltose versus sucrose, if you take into consideration, definitely it will vary from species to species, genotype and many other factors. But yes, because of these properties, maltose can be considered as a very good alternative for sucrose. Then regarding agar concentration, as I told you, if we are reducing the agar concentration, it will weaken the gel structure. increase in the fluidity, and obviously the structural support will be reduced. So this will in turn leads to affecting the morphogenesis, growth and development. Therefore, it requires careful optimization to ensure successful tissue culture outcomes. Moving on to our third question, that is regarding transgenic animal. The transgenic mouse and some statements regarding the same is provided over here. The transgenic mouse thus born will be homozygous transgenic animal and can be maintained by crossing with another transgenic animal. Fertilized transgenic eggs are allowed to develop under in vitro condition. The desired gene is preferably micro-injected into male pronucleus after the sperm entry in the oocytes. For best efficiency, the desired gene is always micro-injected in the male gametes and then they are allowed to fertilize the female gametes. Blastocyst stage embryos are transferred to the uterus of hormonally prepared mother. The fertilized eggs are collected from the specific strains of mouse. The female mouse of specific strain is superovulated and oocytes are collected and allowed to fertilize under in vitro condition. Now, in this case, you have to identify the correct sequence through which a transgenic mouse has to be generated. So, the correct answer over here is G, C and E. G is the female mouse of a specific strain is superovulated, oocytes are collected and allowed to fertilize in vitro. After that, it is micro-injected into the male pronucleus. Then, the blastocyst stage embryos will be transferred to the uterus of an hormonally prepared mother. So, in this case, what we need to remember is that we have three methods by which a transgenic mouse can be generated. First is establishing the transgenic mice with retroviral vectors. So, you are taking a donor female, extracting the 8-cell stage embryo. We are taking a vehicle, that is retrovirus, that carries the transgene, inserting into that. So, this implanted female upon fertilization will give rise to the pups. Whoever carries the transgene is considered to be a transgenic founder from where the transgenic line can be established. Second method is establishing the transgenic mice by means of DNA micro-injection. That means we are taking a super-ovulated female. That means which can synthesize eggs more than the extra number. We are taking the male pronucleus and female pronucleus. Holding with the help of a pipette, we are injecting the transgene into the male pronucleus, transferring into the implanted female. And upon fertilization, it will be producing the pups. Whoever will carry the transgene is known as transgenic founder, from where the transgenic lines can be established. So, the third method of producing transgenic mice is using genetically engineered embryonic stem cells. So, we are taking a donor blastocyst, embryonic stem cells, and transgene is given, or transfected we can say. So, what we get embryonic stem cells, transfected embryonic stem cells. Now, these transfected embryonic stem cells will be micro-injected into the recipient blastocyst, implanted into the female mother. Upon fertilization, it will give rise to the pups. Whichever pup will be carrying the transgene becomes a transgenic founder, from where the transgenic line can be established. So, this is one of the important topic when it comes to CSIR point of view. So, I hope so I gave you some idea about the topics from Unit 12. I will definitely come up with another session where I'll be discussing few more topics and the relevant questions. Thank you.